So first, I'm really happy to be here, and so first, let me introduce myself. So I'm a professor in the physiology and biophysics department at one of the large medical centers in Chicago. And for pretty much all of my career, I've been doing modeling of biophysical stuff. And I guess the two common themes are ions moving somewhere, and also that I'm not using like standard packages, and I don't do like molecular dynamics. So it's always been, you write down some sort of physics, and then you have an equation, and then I need to solve it. So it's that kind of scientific computation that I've been doing. And so it's a lot of, besides the physics and the biology, it's a lot of algorithm development, a lot of programming. And so what I wanted to talk about today, so I was asked to bring up something that comes out of my research. And this is like brand new stuff that I'm doing right now and that I'm using right now. And it's an algorithm that I developed just like a couple years ago. So it is, at least for me, new. It is not earth-shattering or anything, but it is a good, solid algorithm to do what I need to do, and we'll talk about all the specifics of that. So what I want to make clear is that I would, if you all have questions, just throw them out and be interactive, because me talking up here for 6, 10, 12 hours, whatever it is probably not the world's most exciting thing.
But there will be also a hands-on part.
Yeah, yes.
So you need to program something.
Yes, you will need to program something at the end of the day, and it's actually sort of quite ambitious, so I want to, so we'll see how much we can get done in that part. But like I said, if you have questions, just throw them out. I'm not totally sure what everybody's background is, so if I say something that you don't know about, or if I lost you somewhere, please speak up. So I wanted to not only talk about like a specific algorithm, but also sort of things I've learned along the way about being a scientific computation guy. And so I want to sort of start and end today with a guide for succeeding in modeling, at least in biology. And there's just sort of a few things that, some of which we'll expound on in more detail, but that I've sort of learned that make you more successful. So first, know the biology. This
could be applied to anything.
Yes. If it's physics, yes. No, but like the reason that I put this in, especially if you're somebody who's just studying how to be a simulator, it's important to know what is going on and how the experiments are done, because it really helps you not only understand what the actual sort of set of parameters are that you're working with, what is a large number, what is a small number, but it also helps you understand whether you just computed something stupid or you computed something smart. Because it could be, if you don't recognize that you made an error and you created new physics because you had a typo somewhere, it's important to know these kinds of things. And the biology or the physics or the chemistry really helps you ground yourself, and it sets the basis for where you want to be. Because the equations, because I've worked with mathematicians, and they're like, the reaction diffusion equations or the whatever equations, they're just equations, and everything else is numbers. Numbers is numbers. And numbers is not numbers, because everything in whatever world you're working in takes a certain range. And so the parameters may go from 10 to the minus 6 to 10 to the 6, which is a really big range, but it doesn't have to go to infinity. So your code has to fit that range well that you're working in. And it doesn't have to fit all the numbers, which is how mathematicians think. And that's the kind of stuff that knowing the biology and the physics and the chemistry really helps you with. Second, and this is more towards like writing your own code and doing scientific computation. The more you tailor the solver to your specific system, the better. So if you're going to solve certain equations, like today the reaction diffusion equations, the more information that you put in about your system, the more efficient you can make it. It's essentially it's the difference between a black box solver that knows nothing about your equations and your system, or adding more information lets the algorithm be more efficient. And we'll go over some examples of that today. But basically, you're adding more information, and that's always better. You don't need a Ferrari. Try to do better than a Honda. Aim for a Porsche. So we'll talk about this in more detail, but basically what this means is you don't need something extra super fancy that is really expensive and that is finicky and breaks easily. But on the other hand, Hondas are great. Nothing against the Honda Civic. It's a great standard. It'll get you where you want to go, but for what you all are doing as professionals, it's a little bit beneath you, and you can do better. As in real life, most everybody, hopefully, with a good job, can afford a Porsche, a Honda, but with a little bit of hard work and some effort, maybe we can move up to a Porsche. And that's what we're aiming at today. One of the most important things is check, check, check. Check your code, and most importantly, try to break your code. Like, push it to its extremes, which I have found accidentally is one of my superpowers in life. If somebody gives me their code, whatever I want to work on, I will probably find a bug and break it. And this is just important for life, and it gets into the next point: know the performance envelope of your simulator. So for what conditions does it work well? For what kind of parameters that you have for your equations? Like we have reaction diffusion equation that has a bunch of parameters in it. Diffusion coefficient, the on rate, the off rate of the reactions. These are all, in principle, just numbers in front of some of the terms. But if you push them to really big numbers, your simulator may not be able to handle that well, or to really small numbers. And so the idea of understanding the performance envelope of your simulator, know when it fails, know when it works well, so that you can avoid being embarrassed when you find out that this exciting new result that you just showed your boss is actually just garbage in, garbage out. So that's sort of the beginning. So let's start by knowing the biology of what we want to talk about today. So this is a cardiac myocyte. It's a heart muscle cell, and it has been loaded with dyes that fluoresce when they bind calcium. And so the kind of experiment that we're looking at today looks like this. So there's a bunch of things that light up, and those are called calcium sparks. And that is today what we're going to write code to try to model. And so let's go into the biology a little bit. So here you have that same cardiac myocyte, and on the inside of that, running down the long axis here, you have these myofibrils. And so these are these long tubes, and each one of them is wrapped in a network of two different kinds of tubules. And one of those networks will be of interest to us. So here we have a little blow up of this, and we have the what are called the T tubules, which are the magenta ones, and the sarcoplasmic reticulum, or just the SR. And this is the sarcoplasmic reticulum is, its job is to store calcium inside the cell. So it has a high concentration of calcium inside. That calcium comes in there via pumps. These are proteins that use ATP to pump calcium against its concentration gradient into the SR. And the part that we're interested in, these ryanodine receptor ion channels, are right at the junction of those two tubule systems. And so this is actually a skeletal muscle cell, and we're interested in the heart muscle cell. And the one place where they are different is actually at this juncture. So here's a cartoon of that junction in cardiac. So you can see why I used the skeletal muscle picture, because it's a much higher quality. So here we have the T tubule, which is the magenta part, and then we have the SR, which is this blue part, the same blue part here. So in heart, there's only a single sort of point of contact, a flattening of the SR against the T tubule, and that's where we're focusing today. And what happens there is you have these ion channels, which are these little squares here, which are called ryanodine receptors, and their job is to let out calcium out of the SR during a muscle contraction. And when you see a spark, that is the release of calcium from one of these junctions. So if you learn nothing else today, here's a fun bit of biology. During every heartbeat, you have the ryanodine receptors opening, dumping a lot of calcium. That calcium goes inside the myofibrils, where the contractile proteins are, and then the contractile proteins are two proteins that go past each other, and then they're connected to the ends of the cell, and so they contract. In order to make that happen, you need calcium at every step to do that, and that's why the calcium is released. And then the calcium is pumped back into the SR with ATP-based pumps, and then the whole cycle starts over again. So this is every heartbeat. You have this calcium out, does stuff, gets pumped back in. And so like in a mouse that has a heartbeat of like 600 beats a minute, this happens all the time as well. So here we're going to be focusing on this. There's a little tiny cleft. So if you have the SR membrane and the T tubule membrane, they're just slightly separated from each other. They're not actually touching. They're very slightly separated, and that's a place called the dyadic cleft. And that's the part that the calcium will diffuse into and then come out into the rest of the cell. And so here is a mock-up from what I have in my simulation. So the pink is the T tubule, the cyan and the blue are the SR, and the little dots are where I have placed the ryanodine receptors so that they release, that's the location where they release calcium. And then just to sort of get an idea what the scales are in this, you have this little tiny it's like 12 nanometers tall, this junction. And then calcium comes out of those locations there and then goes both out the side and out the front of this and diffuses into the rest of the cell. So that's the kind of system that we're studying today. or that we're simulating today, and how does one, what actually happens? So if you look at the very top of the SR, right where it meets the T-tubule, so you have the T-tubule, the SR sort of cups around it a little bit with that small gap, and then underneath there's another part of the SR that sort of feeds into the rest of the system. And if you look at it from on top, the ranitidine receptors are packed together in an array. So it's a regular array of ranitidine receptors. And then what happens is a positive feedback mechanism. So if the heart is at rest, in principle nothing should happen, but sometimes it does. And what happens is that a ranitidine receptor can open spontaneously, it can open randomly. And what it does, it then releases calcium out of the SR, and the way that these channels work to release calcium is by a positive feedback mechanism, in that they not only conduct calcium, but they are also sensitive to calcium, meaning that they open in the presence of increased calcium on this face. So they release calcium, and then their neighbors can see that calcium, and then that might activate that channel, and then that can activate other channels to release. So it can be a positive feedback mechanism. It's called calcium-induced calcium release. So if you have an increase of calcium on this in here, then the channels can all fire, or just a couple channels can fire. So what happens then to get this kind of experiment? The cell has been loaded with calcium fluorescing dyes, and so if we have a protein or a molecule that binds calcium like this, and here's the calcium, they can react in a chemical reaction with an on rate, and then that chemical reaction sometimes reverses itself, and the complexed calcium and buffer can undo itself and become these two separate molecules again. And that's the reaction part. And so you have the diffusion of the calcium throughout, and then the reaction with buffers. There are both ones that you put in to make things light up, but there are also just a lot of proteins inside the cells that bind calcium and that we're going to be modeling. And so this is the reaction part of this system. And what you measure is, so the way you measure this is in the cell, you shine a laser down the long axis of the cell, and you record the amplitude of the light signal that you get, and then you do this over and over and over and over again, and you get a signal that looks like this, where like this line is like this part of the of this line, and then you stack them all up in time. And so this is from second 13.3 of my simulation to the 14th second of my simulation. And you record in time the amplitudes. And if you just look on this line, that's as if you held the laser right at that point all the time. You get a signal that looks like this. So you look at the spark amplitude. And then in the simulations and also in some experiments, you can see what the concentration inside the SR is of calcium. So it starts to get depleted of calcium, and then the pumps start to pump it back in, and then when you get another release event, it depletes again, and then when the channels close, it starts to get filled back up with calcium, both from the pumps and the rest of the SR network. So here's a movie of one of the simulations. So what you have here is you have the T-tubule sort of coming out this way, and then you have the SR wrapped around it, and then this view is just like from on top here, so you're only seeing the very top of these two parts. And then we're going to look at the amplitude and the calcium in the SR. So here at the very top of the spark, at the highest amplitude, you have a bunch of channels open. You have a very strong signal because you have a lot of calcium that's being released. But then as we go on, these signals become weaker because while you still have a lot of channels open, but you don't have very much calcium coming out of it because there isn't very much left. Then the channels start to close, and then the SR starts to refill. And then what happens is something that's really important, which is that nothing happens. If this is the heart at rest, not much should happen. There's some calcium leak, which is normal, like small little blips like this, just calcium leaking out of the SR, that's normal. But while the heart is at rest, not much should happen, which is why sparks are rare. And then we're just going to leave it with in the middle of another sort of big event. And this was just over a half a second of simulated time. So you can see that sparks don't happen very often, which is a good thing physiologically. So you see the calcium diffusing out the front and the back here, and which is sort of coming out this way and that way in this picture, and then spilling over the sides and into the rest of the cell. So that's the 3D version of this. So how do we get this? Now we're getting into the actual math. So we have a few molecular species, obviously calcium, and a bunch of buffers or calcium chelators. Some of these are the fluorescing ones, and some of them are just natural proteins. So we're going to call the calcium concentration C. This is the free, the one that hasn't been taken up by buffer. And it diffuses within the cell. It moves through the ranitidine receptors, and it gets pumped back into the SR from the cytosol. So we have a bunch of buffers. I think in the actual simulations, close to 5 or 6. And so they have different indices, and we're going to look at the unbound concentration. So the ones that are the concentration of the chelators that are available for binding. And then if you want to know how much of it is bound, that's the total, which is a fixed amount that you put into the experiment in the beginning, and then you just subtract off the unbound part. And then this is the chemical reaction again. So you have a forward, an on rate, and an off rate of the calcium unbinding. And then the equations that govern this are these. So what this says is that the rate of change at a location X of in this case, the calcium concentration, is a diffusion part. So just how quickly it diffuses away from that spot or into that spot. It has a source. So this is just a source of calcium that comes into that spot. It can be like the pump as it's moving calcium through one membrane into the SR. One can potentially model the ranitidine receptor current that way, but it's basically just as if you turned on the faucet of the sink, that in that part there's just a constant current of calcium coming in. And then we have the reaction terms. So the concentration of calcium will decrease if it is bound. So this is the on rate, and this is the term that is used to describe the on reaction. And then this is the term that describes the off reaction. So the off reaction is just depends on the concentration of how much of this stuff is there, whereas the on rate depends on both how much buffer there is available and how much calcium there is. And then the buffer concentration is exactly the same. The buffers will diffuse. There are no sources of buffers in this kind of system. There isn't buffer magically appearing, whereas for the calcium you do have these sources that are not diffusive, where calcium comes through the pumps in a way that isn't diffusive and it isn't a reaction. But there's nothing like that for the chelators, and then they decrease, the chelator concentration decreases for the exact same reason that if it binds calcium, this is gone and becomes this, and then it unbinds and increases the concentration for that. So those are the base equations that we're talking about today, and that's what we want to solve. All right, to do that, we're going to first start with the method of lines. So the method of lines is that you have a complicated PDE, and that is time and space dependent, and you coarse grain, you discretize the, in this case, the space element, and you keep the time element so that you wind up making a whole set of ordinary differential equations that are just in time. So we're going to break the entire geometry into little voxels or voxels, or what I call nanoboxes. And so in each box, what we're going to say is in each box, we're going to have one concentration of calcium, one concentration of buffer, and so that we're discretizing everything in space, and then we can have a source from one box to another box, and then they diffuse across the boxes, so that we get a reaction diffusion equation for each box, and we're going to label each box with indices like I and J. So everything here becomes now a set of ordinary differential equations that are just in time, and that are then the space has been discretized into and is represented just by these indices I and J. So where does this come from? So for these kinds of things, if we just say that each box has a concentration, we see that this is the concentration of box I, concentration of box I, and so then these are fairly straightforward. And so where did we get the this diffusion thing with this kind of complicated new diffusion coefficient kind of thing? So this is an application of the divergence theorem. So if we just say we're gonna average over the volume of each box. So here we take this term and do the average value over this box. If this is this box has volume VI, we integrate this, and then using the divergence theorem, we just get the flux over the boundaries, and then we know how to generally do that. So here the gradient is then approximated by a difference, like we normally do for a derivative. Instead of doing a derivative, we take the value here, we take the value here, and divide by the distance between them. And this is the concentration in box I, concentration in box J, difference over that divided by the distance between the centers of the boxes. So that's this gradient, and then we get the V from here, and then the A comes in because we're doing the surface integral, so we need the area of the common face. That's where this new diffusion coefficient comes from. And these are this is usually the first kind of step that you need to do, because there's a lot of techniques for doing systems of ordinary differential equations that are a lot easier to deal with after you discretize the Okay. This is just a difference divided by the length, and then because you're doing surface integrals, you're always going to have to multiply it by the available surface area.
Are you familiar with math?
Yes, no, I was just wondering, because there's the Laplacian on the left-hand side of the equation. There are also two gradients on the right-hand side.
Yeah. So this then is the flux. So we're just taking basically the number of lines that come over here, and we're just gonna, because everything is sort of uniform in each box, like everything is a constant concentration. It's literally just the difference between the two, the flux. You always want the one that is perpendicular to the. What you're looking at is the amount of flux that is coming through that is perpendicular to the wall. So in this case, everything is perpendicular just by the way that it's uniform, and I don't know how better to describe that. But this is a quite common thing to do. It's let's leave it at that, and maybe we can talk about it in the break if you want. But it's a very common thing to do in physics, and to apply it this way. It's just a matter of figuring out what the flux is, and in this case, it's.
Yeah, no, I'm just wondering the approximation below the second row. This is for the first derivative of the concentration.
Right. That's for this. And then the surface integral, the flux integral, which is includes this part, including this del, is the area, because all the flux happens to be perpendicular to that area. Okay, so what are the challenges of modeling sparks in this kind of system? So first we have a system of nonlinearly coupled PDEs. And it's nonlinearly coupled because you have this product of the unknowns that you want. And so that's one complication that we're going to need to deal with. There's a huge difference in timescales. So whenever you have these ranitidine receptors opening, you're dumping a lot of calcium into a very small space in a very short amount of time. And so what you need is basically submicrosecond resolution in your model, because it's a very small cleft that you're going into, and the channels opening make a sudden change in the calcium concentration. Then there's the diffusion into the cell. That takes about 10,000 times longer. So is on the millisecond time scale. The sparks themselves will last 10 to 100 milliseconds. So, and then the experiments are tens of seconds to minutes. So you're covering 8 to 9 orders of magnitude in all of this. And you need to simulate all of that because you essentially want to reproduce the experiment. Then there's also really large differences in length scales. So the diatic cleft is like 12 nanometers, whereas the rest of the cell is microns, so it's thousands of nanometers, and a spark itself can get several thousand nanometers wide. Really? Really? All right. The other really challenging part is that sparks are rare. If you actually want to study sparks and get basically enough statistical data to do an analysis of sparks. You get a spark like every second, maybe two a second in your simulations. And so you need a lot of simulations over a long period of time in order to get enough data to do that. And how do we deal with this? So first, for the geometric part, so this is a
We could add that this is really a general feature of biophysics, biological. So that,
it is very general.
Rare.
It is a very general problem,
So
yes.
we have noise, and then some signal starts, and that's besides the complex in the system, you have also complex in the timescales as well, because you cannot just run your simulation and wait. I think these are very general.
Oh yeah, these are very.
I think it's the key problem in biology, right? You're dealing with rare events all the time.
Yes, you're dealing with rare events all the time. You're certainly dealing with different length scales all the time, and different time scales. This is probably a fairly extreme difference in time scales, but anytime you're looking to get to the experimental time scales, you're always looking at seconds and minutes. This isn't physics where you're looking at like attoseconds, but rather biological experiments are always seconds or minutes. And all the interesting biology happens on the millisecond time scale, so at the very least. So this is a simulation box. This is about half the size of what I normally use because it allows you to see it better. So this is the pink is the T tubule, and then the cyan and the blue are the SR sort of wrapping around it. And the point of this is that we are using very small boxes where the action is, and that's right in here. And here you see a slightly different version of the same thing. So here we have a bunch of, in the red spot, which is the dyadic clef, we have very tiny boxes, and then the boxes get larger as we get farther away, and things change less rapidly. Here, even if something is going on rapidly here, and we can coarse grain essentially these things out here. And so that's the easy fix for dealing with length scales. Time scales are a whole different matter, and that's what we're spending most of our time worrying about is how to deal with these different time scales. All right, so if these are the ODEs that we want to solve, what are some sort of standard ways to look at this? These are generally the Euler solution methods, and so here we're going to use throughout we're going to use a shorthand notation where we shortcut all of this and say that it looks like this, and all of these things, the U and the F, are vectors. So to make a system of equations, you have all your unknowns that are dumped into this one variable, and then this F is also a matrix sort of function to get or a vector where each element is essentially this. So then the sort of standard methods for doing this are forward Euler, backward Euler, and Crank-Nicolson. So forward Euler is the simplest one, and as you'll find out, it's the one for which I have the most disdain. Is you discretize the time derivative with a one-sided derivative, and so here un is the u at time n, and then so this is u at the next time step n plus 1, and then you divide it by the length of the time step, and then for the forward Euler you just put in f of the initial time step. So solving this is really easy. So forward Euler has the great property of being really easy to solve. So if you're what you want to solve for is the next time step, so you want to solve for un plus 1. Well, that's just real straightforward here. And so you can just use it quickly over and over again. But if the other thing you should learn from this is please don't use forward Euler. Backward Euler. Forward Euler is, as you hopefully know, unstable and will give you really garbage answers if you don't pick your h small enough. Backward Euler is now the exact same thing, except here in your f you put the u that you want to find, so the one at the next time step. So this then makes it more difficult to solve. So for us, that involves a nonlinearity. So if everything was just linear, if it was just the like the diffusion part, we could just move all the u's over to one side, and you would make a matrix equation and solve things. But here, for our case, we have to solve a nonlinear equation, which is not straightforward and requires work. Backward Euler is, however, stable and will be wrong in a less dramatic sense than the forward Euler. And then Crank-Nicholson is the average of the two, and so you're still having to solve a nonlinear problem. All right, so those are sort of the normal sort of standard ways, and I wanted to include it because we're going to be doing some backward Euler as part of the operator splitting. So for the operator splitting, we're going to get into all the details. But basically what we have is two parts on the right-hand side. For our case, it's the diffusion and the reaction. But it's a very general method, and you can use it for pretty much anything that splits up into an additive part like this. So just the very basic idea of operator splitting is that it is much more, if you can much more efficiently do each part separately, if you can solve this equation very efficiently, if you can solve this equation very efficiently, but together you can't do it efficiently, then the idea of operator splitting is to solve these two things separately, somehow combine them into the answer that you want for the actual answer of the whole problem. And that's the most general idea of operator splitting. All right, so now I want to talk about like general methods, classes of solution methods for basically systems of ODEs in general. And this is not an official sort of class. This is just sort of how I think of it. So you have your tractors, you have your Honda Civics, you have your Porsches, you have your Ferraris or your F1 cars. And for the favorite of many biologists is the Ford Pinto. Most everybody here probably doesn't remember the Ford Pinto. It was a car in the 70s that had this unfortunate habit of exploding if it got into an accident, even a relatively minor accident. And for some reason, biologists seem to really love this because I don't know why. But so what's a tractor? A tractor is one of those generalist black box packages that come in MATLAB or Mathematica or something like that. A lot of them are based on the old classic Runge-Kutta methods, and they don't know anything about your equations, but it will get you there. It will get you there slowly, but it will roll over everything in its path and probably get you to a solution. Your Honda Civic is also generalist, but it's more tailored to your current problem, and a good example of that is the backward Euler. So you have to solve some equations to do the backward Euler, and if you have a nonlinearity, you have choice as to how you approximate that or how you solve that. And so there is some tailoring to whatever problem, whatever system of equations you have, because you have to make decisions as to how to solve that backward Euler equation. Now the Porsche is also still a generalist. It's a general sort of car, but it's much more sophisticated and has a few more bells and whistles, and it's much more tailored to your current problem. And an example of something that we'll be doing that is Porsche-esque, it would be like dynamic time stepping. So you're not just taking the same uniform time step all the time. And then your F1 car, your Ferrari, is highly tailored for really complicated problems. In physics, especially CFD, computational fluid dynamics, nuclear physics, those require really high accuracy and really fancy code. This is really difficult to code to build, to do the theory for, and it tends to be very tailored to the problems that you're trying to do, and it requires way too much work, at least for what I want to do. And it's generally not necessary unless you're doing these really complicated problems. Now, the Ford Pinto is unstable, it has unpredictable errors, and a good example of that is the forward oiler. If you don't know about forward oiler, if you pick your time step too large, it can do something, it's very unstable, and you will go off to NANs if you're lucky. If you're unlucky, then what it will do, it will just oscillate wildly, sort of about the right answer. But if you're not careful in looking at your answer, you may think, oh, it's fine because I didn't get NANs. And that is why I'm not a fan of the forward oiler.
Just to give you a concrete example about nuclear physics. So the typical thing is to calculate the mass of the hydrogen, whatever, the fifth, 10th digit. And the accuracy becomes so good that they will have it, they will have experiments if you want to compare with the experiment stops before. So it's easy to kneel down and very specific problem to get really right, then you might need codes to 50,000 lines and so on. But the point of it is that for many processes, maybe you will try to use your scientific creativity to solve it in a simpler manner and get already some insights.
Mm-hmm. And like the dirty little secret of modeling biology, if you can reproduce the experiment to within like 10 or 20% of the right answer, you're doing fantastic.
But if you get the number right, you worry.
Yeah, pretty much.
Because
yeah.
The chances that there are some compensational errors is very high. Right?
Exactly. And so a lot of biology modeling is trying to understand the general trends, the general ideas of things that are going on. And so you don't need a super duper accurate code, but you need something that is not wildly wrong. I don't know if that's a good or a bad thing to tell you, but that is sort of the reality of biological modeling. So where does operator splitting fit in? Operator splitting is generally one of these two categories. You can do backward Euler or something simple for each one of these, and so it's more tailored to the current problem because you do have to make decisions as to how to solve things. But we're going to try to work it over here to the Porsche side. All right. So just to give you an idea of if you're I believe you will all be professional modelers. So things you should not do. These are actual methods sections coming out of papers that are in my field doing exactly these reaction diffusion equations, 3D, in the same heart muscle that I'm talking about. The equations were solved with some solving package. Some use some solving package. Solved by the Euler method. This is like, I don't even know what to make of that. It's like, is it forward Euler? Is it backward Euler? What is it? It's probably forward Euler. I will give them the benefit of the doubt and say it's backward Euler, that it's stable. Solved using forward Euler with a variable time step designed to ensure stability. So at least they know it's unstable, but it's it's not much information. Used operator splitting with a fixed time step. So and I want to be clear, these are the entire method sections of these papers, are these one sentences. And one sentence is not a method section. You should not do this. This is what biologists do when they start to code because they're not professional simulators. And the reviewers don't make them expound on this. Like, what kind of checks did you do? All those sorts of things. So you as a professional should do much better. So where do these rank? So we have a couple of tractors. They used packages. I will give we use the Euler method, the benefit of the doubt that they use backward Euler. And then we had at least one Ford Pinto with using the forward Euler. So this is generally where biologists wind up doing simulations. Those are the kinds of methods that they use because they're not professionals at this. Simulation and modeling is not something that is always thought of as, it can be thought of as doing something you can just do on the side and it doesn't really require that much expertise, which of course is not true. So what are our goals today? Our goal is to make a Porsche. And so I want to describe operator splitting. I want to see how we tailor it to the problems that we have and how we do the dynamic time stepping. Then in the second session this afternoon, it's hands-on trying to do some stuff like this with numerical examples. MATLAB, Mathematica, Python are probably best for doing this, although you can use other stuff if you want. If you use Mathematica, you can copy everything just off the board from what I'm going to show you, because I'm going to show you my Mathematica code. So if you want to make life easy for yourself. All right, timescales. So we need to worry about how to do different timescales. So here we have our whole simulation, and we're going to call the whole simulation time capital T. And then we're going to divide T into delta T's, and then in each delta T interval, the ranitidine receptor currents are going to be constant. So what's happening here is that a ranitidine receptor opened, and then, or at least one, maybe two, and then their current is constant for the entire time. All the ones that are off are not conducting current, they're off. And then here, at this time, at this delta T, we ask the question, do the open channels want to close, and do the closed channels want to open? We flip the probabilities of that, which is part of the modeling. And then during the next delta T, all the channels that are open have a constant current, which may be smaller than the one before because we've depleted some of the calcium. Or in this case, it's probably always the same. And then we have our delta T here, and it is in this delta T that we're going to use the operator splitting to solve the reaction diffusion equations, and hopefully our goal is to do a non-uniform time step so that when a channel has opened, we use very small time steps, and then as things get closer to steady state, when things are changing less quickly, we can use larger and larger time steps. And we're going to call those H. All right, the things that we have to worry about. There's at least 4 different kinds of error things that we need to worry about. The first one is local error. So how good is my operator splitting on this one interval that I'm using it for? And then basically how accurate do I solve this one little thing? Then a global error over this entire delta t is if I do many h intervals, how good is my answer at the end of it all, at the end of that. So if you have small errors or large errors in your h, then they can accumulate and make big problems at the end of your delta t. Then we want to string together a bunch of delta t's to make my whole simulation. So then there's stability. At the end of my really long cap t simulation, which for what I'm doing is about 15 seconds of simulated time, is that answer accurate at the end of it all? After I've strung together a huge number of h1s and a large number of delta t intervals, has error accumulated throughout all that time, or is the error sort of drifting off and becoming larger and larger? And then there's the idea of consistency. This is what we're going to come to a few times. As h goes to 0, as you make these intervals smaller and smaller, do you get a more and more accurate answer? And operator splitting has been shown to be consistent, so we're not going to worry about this. We're more going to use the fact that operator splitting is consistent to help us deal with and control these errors. So these are always the things you need to worry about. You're always going to be doing something on one small time step. How accurate is that? And then if you string a bunch of these small time steps together, do you have errors accumulating and ruining your entire simulation? So what we're going to be worrying about is trying to control local error, and hopefully everything else will follow. And that's the way that we're going about it. So for the operator splitting, using that same vector notation, we have our diffusive part and our reactive part, and we're going to use the operator splitting over a single h interval. So that means that between these two times, we're going to start at time tn and go to time tn plus 1, which is tn plus h. And then we want to compute u at the end of that interval with operator splitting. And these are systems of ODEs, so you need an initial value always, because any differential equation requires a boundary condition, and for a time-dependent one, that's an initial condition. And so we have an initial condition of u of tn. So that means we've solved the previous time interval from tn minus 1 to tn, and then using that result as our input for the next h. And then repeat. Go on to tn plus 2, and using the initial conditions of the thing you just found, the initial condition for the next one is the stuff you just found at the end of this interval to compute the next one. And we can either use the same time step if you're doing uniform time stepping, or use a dynamic time step where we've increased h based on some knowledge. So that's the general plan. All right, so we talked briefly about somehow we want to we know how to solve this efficiently, we know how to solve this efficiently. Somehow we want to combine them to make the actual answer. Let's flesh this out a little bit for our particular problem, the diffusion. Again, the vector notation. So for the diffusion subproblem, this part is just the diffusive part along with the source. So that we just have this part of our thing, and then for the reaction part, this set of equations would just be all the stuff that is the reaction part. So we have our equations, our initial condition. So operator splitting comes in sort of two forms: Lie splitting and Strang splitting, or first order splitting and second order splitting. If you've studied some math textbooks, some of them maybe by Gilbert Strang, he's written a huge ton of textbooks. That's the same guy. So for operator splitting, we use this kind of diagram to illustrate the ideas. So for Lie splitting, what happens is you do one of these reactions, say, or one of these equations, like the D subproblem for the entire time interval from 0 to h, or Tn to Tn plus 1. And then you take that result, and then you react it. If you did the diffusion here, then you do the reaction, taking the answer at the end of this as the start of this. So what that looks like is we have our first subproblem. That's what this is about. So say if we do the diffusion first with our initial conditions, the initial condition of the problem, and then we solve that over the entire time interval, and then we use that result as the initial condition for the second subproblem, which is the reaction subproblem in this case, and then the answer, or our guess for what U is at the end of this is the end of this subproblem, having solved it over the entire interval. Yeah?
Does this depend on the order? So if you do the first.
That is an excellent question, and we are going to dive into that a lot in this. The order very much does matter, which is one thing I've come to really dislike about operator splitting. It's like it's in some senses very user dependent, and you have to check which is the better order to do things in. So for Strang splitting, it's a little bit different. So first we do one subproblem for half the interval, take that answer, then run the next subproblem for the entire interval, take that answer, and do the first subproblem again for the other half of that interval. So it looks like this. Again, the same initial conditions. Now we only run it for half the interval and use that as the input for this, and then run the, in this case, the diffusion again for starting with this result at the end of the second subproblem. So why, and then the answer is the end of this at the half interval. All right, so why is it structured like this? So let's take a relatively simple system, and we're going to du dt, and in this case we're going to do a system of linear equations. So this is a matrix, but the system's overall a linear system. And I've written it like this because we want to do the operator splitting version of this. So we know what the answer is. We know that it is e to the a plus b t times the initial condition. So recall from your linear algebra or matrix class that the exponential of a matrix is a matrix, and it's defined as being this power series. And so if you have the important part to take away from this is not just this definition, but the fact that this is a matrix, and we're going to use that as we're going to use that fact in a bit. So what happens if you do Lie splitting on this kind of thing? So the solution to the first subproblem is this, and then the second subproblem is e to the b times the initial condition. Well, that initial condition is this. So this is then your answer for your guess for what u of h is. And the important thing is that this does not equal, these two things are not equal because these e to the matrices is a matrix. So these two things are only equal if AB equals BA. If the multiplication is commutative, then you would get that same, then you would have an equality. And to show that, let's use this fact and do an expansion in H of this and of this. And so let's basically subtract these two matrices from each other. And for small h, as you expand here, all the order 1 terms cancel, the order h terms cancel, and what you're left with is an order h squared term. That is exactly this difference. And this notation is the commutator, which you may run across in physics, but it's literally just this definition of what is the difference between A times B and B times A. And since matrices almost never commute, this is not zero. So the idea of the Strang splitting, to do it in this sort of way, is exactly to get rid of this term. So to make it one order better in accuracy, at least for the linear problem. So at the end of the Strang splitting, if you do it all, you get e to the AH over 2, e to the BH, and e to the AH over 2. And this again does not equal this because these don't commute. But if you do an expansion, this term then cancels and you get a third order term. So the difference between these two is one order in H. So so least splitting is a first order accurate method, and Strang splitting, whoops, oh, wrong button. And Strang splitting is a second order accurate method. And it's, at least for the linear problem, so it's specifically designed so that for the linear problem, that term cancels out. And so here's the first time that you see that the order matters. Because if we had done A and B and switched the order, then it would be reversed, but then you would just have the negative of this number. Right? And so what you see is that the answer that you get in switching the order, even here, you get a difference in this error term depending on what order you have, whether you did A first or B first. And so your error will be different, so you know that you have a different answer already, even for this linear problem. You get a different answer whether you did A or B first. So we're going to do Strang splitting in everything that we're doing today. And so this is exactly what I just told you. We do the D, we do the R, and then we do the D. And because we're interested in understanding whether the order matters or not, we're going to call this DRD. And then if we do reaction first, we're going to call that RDR. And then for a bunch of the stuff that we're doing, we're going to compare what the difference is and see which one is better for the problems that we're working on. All right. So these are sort of the generalities. So let's start getting into how we solve the different subproblems, the diffusion subproblem and the reaction subproblem. So the important part of operator splitting is that you solve each of these subproblems much more quickly than you do the system as a whole. And so for Strang splitting, you have to do three things. You have to do, say, D, R, and then D. So you have to do those three things faster and as accurate as if you could do the whole thing by itself, say with like backward Euler. All right, so here's our diffusion subproblem, and the way one normally goes about discretizing this to solve it. So we're going to introduce a new timescale, tau. And tau is just h over 2 or h, depending on which order you're doing things in. So if you're doing diffusion first, then you're doing half an interval. And if you're doing diffusion second, then it's over the whole interval. And so we just use tau as a shorthand for that. And all right, so if we're discretizing this and we're going to solve these using backward Euler, because you know by now how much I love forward Euler. So here we have the unknowns highlighted in red. And so this is just a one-sided derivative. And then we need to solve this. But this is a linear problem. So it's just a matter of bringing all these guys over to the other side and then making a matrix that represents that equation. So you have the identity matrix and plus something that is tau times a bunch of other stuff that is from these guys times your unknowns, your vector of unknowns, and your right-hand side. That relatively clear why you would get a matrix out of this. I'll take that, yeah. Okay, so what have we gained by dropping out the reaction part? We have gained a linear problem. And so all the nonlinearity is in the reactions, because that's where we had the product. And so by dropping out the reaction part and just focusing on the diffusion, we could make a matrix problem that is relatively easy to solve. But it's a special kind of matrix problem because M is very, very sparse. So a box has only up to 6 neighbors. And so each one of these things can only diffuse between 6 neighbors. So if you have, say, 1,000 or 10,000 boxes, each one only has 6 neighbors. That means every row of your matrix has very few elements that are filled in. So there are at most 6 neighbors for each of these guys, and each of these buffers. So it is a very sparse matrix. And unfortunately, it has no specific structure. So if you do like one-dimensional diffusion, what you get is a nice tri-diagonal matrix for which there are special solvers that you can use. But in this case, you almost never have specific structure like that because it matters which order your boxes are in and whether you want to reorganize your matrix in order to make it have special structure. I've chosen not to. I've chosen to order my boxes in a different way so that I could quickly extract the cytosol boxes, the SR boxes, and things like that. That was more important for me. So if you have a very sparse matrix with no specific structure, that basically screams use GMRes. And for those who haven't come across GMRes, which is the generalized minimal residue, it's an iterative matrix equation solver. So it takes a guess and then refines it iteratively. But the nice thing about it is it only uses matrix vector multiplies. You don't actually have to store the matrix, and you only have to tell it how to do the matrix vector multiply, and if it's sparse, that is very quick. All right, so GMRes or solving this system can be a source of instability. Ask me how I know. I didn't solve GMRes to high enough accuracy, and then at some point I saw at the end it started drifting off into something else, and once I made it solve to a higher accuracy, that resolved that problem. So basically you wind up accumulating local errors, so each H interval makes a slight inaccuracy, Those errors accumulate over many delta t's, and over a long simulation, that can become unstable, and the answer starts to drift away from the correct answer. All right, the reaction subproblem is another place where we're going to use, or is a place where we're really going to exploit the structure of the equations that we have. So here we have our equations for the reaction problem, and again we have a tau. In this case, it's h if it's in the middle, or 1/2 if it's done first. And all right, so now these are the equations that we need to solve. And now we have a linearity, a nonlinearity. But we have also made this very advantageous for us by removing the diffusion part. So the biggest one is that there are no neighbors involved. So all you see here are I, box I, box I, box I, box I. There's no neighbors involved. So that means that everything that's going on is happening just inside each box, and each box is independent of all the other boxes. So there's no neighbors communicating. And whereas like the diffusion goes from one to the other, here we're just doing everything inside one box. And this, and we can also exploit the specifics of our system for this. So first, and we're going to go through this in detail, but we only have one species that is chelated, that's the C. That's one thing that we're going to exploit. And the fact that the C and B equations are very similar. So if you look, the C equation is basically just the sum of the B equations. And the nonlinear coupling is much easier to deal with in this. All right, so now I've simplified the notation a little bit by getting rid of the i's and just by looking at C and B as our unknowns. And then it's just each box has its own equation. All right, so we would like to solve these equations, but it's actually simpler to exploit the structure of these equations to solve these equations. So here, this is what I just said. This equation is the sum of these equations. And we can integrate this exactly. There's no approximation here. And we get this. And this becomes, in this notation, becomes just C equals the sum of B's, essentially, plus some initial stuff. And this equation is much more useful than this equation. And we're going to then use this equation. We're going to rewrite it in a second and combine it with this one. So here, we can rewrite this equation and solve it for B in terms of C. So that's just rearranging these terms and grouping a bunch of terms together. And then I can substitute this in here and come up with one equation to solve that is just in terms of C. There are no B's in here at all. And so I have one equation for every box that I can solve for C. That is, it is a one variable nonlinear equation, but it's a pretty simple formula. And I need to solve it to high accuracy, and so that screams Newton's method. So you have a single equation, a nonlinear equation that is solved to high accuracy, Newton's method, especially by a simple formula. Just to recap Newton's method, if you want to solve f of x equals 0, it's iterative. So your next guess is your first guess minus f of your guess divided by f by the derivative. And then in our case, f has these simple formulas. And why do we want to use Newton's method? Because of square convergence. So every time you do an iteration, you basically double the number of significant figures that you get in your answer. And so for our problem, we generally only need 1 or 2 iterations, so it's really fast. And all operations can be done vector-wise. So this equation is for box 1, for box 2, for box 3. We have the same exact equation, so we can just line up all our equations and do all of this stuff vector-wise, and that's really fast. So what do I mean by vector-wise? So let's see about like this denominator, how I would calculate that. So I would make a vector of all my combined capital B's for each box, and then my X in each box, or which would be like the C in each box. And then you just add them up component-wise, and everything is done component-wise. And so if you give MATLAB or Mathematica this vector, it will just add everything vector-wise, and it will divide everything, element-wise, and it will divide things element-wise. And so you just have one string of stuff that you're doing all of this to iteratively, and it's really fast. And that's why the reaction subproblem can be done fast. And then to get the B's, you go back to that other equation and just put in the answer that you had here, and you get the B's. And so what we've done here is exploit the structure of our equations so that we can solve this more quickly. Oh, wow, I actually got that done right on time. So this is what I mean by you're adding more information by using the structure of your equations and what is specific about your equations to do something better. If it was a black box, it wouldn't know to take these kinds of shortcuts, and that is why this works for this particular case that we're looking for. And that's why I mean by that's what I mean by tailoring things to your system. And so that's the basics of operator splitting. And then in the next session, we're going to talk about everything that can go wrong. So let's take a break for a half hour and then come back. Let's keep going here. All right, so clearly operator splitting is great. It's easy, but that's probably a good title for this session. But just a reminder, consistency is as h goes to 0, the solution becomes more accurate. So that's known for operator splitting, but I want to examine that a little bit more. So there is a very real-world issue, even for the most simple system. And so let's take a look at literally the most simple system. So before we had a system like this, but there were matrices. These are just numbers. So we just have one single ordinary differential equation, and that is linear, that we want to study with the operator splitting. So we know the answer for this kind of differential equation. It's like the first thing that you learn in the differential equations class is that the answer is this. So for this particular one, with all the A's and the B's, it's like this. And we're going to assume that A and B are negative so that it has a long-term solution. Otherwise, it goes off to infinity with this term. So then that long-term solution, that steady-state solution, is just the limit of this with this exponential going away, so you're just left with this part here. And the other way to find that would just be to set this part equal to 0, the derivative is 0, steady state means that you're not changing, and then you can just solve that for u and you get this. So far so good. So that's all pretty straightforward for the actual answer. But what does operator splitting give for the steady state? So if we apply the Strang operator splitting to the system, the first answer is this. Put that into here as the initial condition for the solution to the second system, third system, and then the answer at Tn plus 1 is at the end of this. So how do we get the steady state solution out of this? That is, at steady state, Tn plus 1 is supposed to equal Tn, or sorry, U of Tn plus 1 is supposed to equal U of Tn. It's the definition, the derivative is 0, so it's flat. And so what we can do is to set these two things equal to each other, solve this whole system. It's really straightforward, and you get that. So this clearly is not that. So let's take a look at that in a little bit of detail. And we can do an H expansion of this. For small time steps, we get our the actual steady state answer plus an H squared error term plus an H to the fourth term. So what we see here is that yes, operator splitting is consistent. And as H goes to 0, you get to the right final steady state solution. But it does create a problem in that even for this really simple case, you can't use large time steps. And so that, because you're not going to get to the correct steady state solution if this is anything close to order 1. And that is going to be a problem because what we want to do is do the dynamic time stepping, where we're doing first short and small short time steps, small h's, but then we get bigger and bigger and bigger ones. And so that is going to be a problem for us. And the technical way to describe this is that operator splitting does not preserve the steady state. So for the steady state, you get a different answer than the actual true steady state answer. And we'll explore that a little bit in the workshop. All right, so how do we try to overcome that? And that is something called balanced operator splitting. The idea of balanced operator splitting is to really not change the equations that we have, but to add a term and subtract the same term, so that overall the actual equations, the actual equation that we want to solve is unchanged, but then our subproblems become slightly different. And so the idea is that you're adding a little bit of flux to the diffusion part, and then you're decreasing the rate of reaction a little bit. And the idea is to try to find a psi that gets you to the right steady state. And so then the subproblems just become plus the psi, minus the psi, and then the d plus the psi again. And then that is your updated answer. And so what is the psi that you actually want to put there? There is actually a psi. So if you know the answer to your problem, if you know the steady state answer, then the thing that you want to add is this. So R at the steady state minus D at the steady state and a half of that. So why do you want to do that? Why is that the term? And I'm not going to go through all the exact details, but I think you'll see the gist of it. So at steady state, where du dt is 0, we have this relationship. Now, if you start your first subproblem with an initial condition of it being the correct steady state that you don't know, but if you did, if you started it there, then you can prove that V1 is actually constant if you have this psi. And the reason for that is if this is your psi and this is your first subproblem, if you put in u infinity in here, then that just becomes this. So you're adding half d, subtracting off a half a d, so you get a half a plus d, and we know that this is 0, and that's use that fact for the proof that the first subproblem is actually just constant at that steady state answer. And then that means that the second subproblem will also be constant and it's at steady state. Then the third problem is as well. So that then so that means that at the end of it all, you get this number back that you started with, and so you're in steady state and you're at the correct steady state because this is what you put in to begin with. So there is at least something that you can put in that guarantees that you get to the right steady state. Of course, we don't know what that is, but at least this shows you that there is something. And then there is a way to guarantee to reach the correct steady state. Now the question is, how do we implement that? Because we don't actually know this number. All right, so the first obvious thing to try would be to say that at time step n, and since I don't know this, I'm just going to put in un and do it this way, and then update my psi at every time interval. And that, unfortunately, is unstable, even for really simple linear systems. And it's only stable for small h. So it's, again, not something that we want to do. So what Speth et al., which is a paper that includes Gilbert Strang of the Strang splitting, came up with is a way to do this that works in practice. So the idea is, instead of putting in this thing that you don't know and not using this thing that doesn't work, the idea is to use the average of the R minus the average of the D at every time step. And then you update it for different time steps, and that is something that actually works in practice. So let's take a look at that. So here we have our DRD operator splitting, and if I just take this equation, our first subproblem, and I just integrate it exactly, then you get this with this undetermined integral d of v1 over half the time interval. And then I can do the same thing for the third time interval, doing the integral over the second half of the time interval, and do something similar for the second subproblem. So this is all just formal stuff. This is just rewriting this equation in an integral form. And we don't necessarily know what this is, but we're going to take this stuff and use these integrals to approximate these integrals. So what's the difference? Here the difference is that you're integrating over u, and here you're integrating over v2, your second subproblem answer, and here you're integrating over your first subproblem, and here you're integrating over your third subproblem. So the idea is that these are close enough to your answer to approximate this, and you know how to compute this. It's really cheap because you have to compute it along the way anyway. So you rearrange these equations to solve for the integral, and you get these things that you're already computing along the way anyway. So you wind up with this guy a couple of times, and then with this guy, and this over here. Actually, that probably should have been an h over 2. And you just combine all these things, and you get this. So what you get is a very simple formula for updating the psi at every time step. There's no proof that it is stable, even for a simple system like a linear system. There's no proof that it's stable, but it seems to work really well in practice, both in the examples that they used in their work and in the stuff that I've been doing. So it seems to work in practice, and it's also very cheap. So it's cheap in two ways. So first, you're already computing these things along the way. So this is the initial condition, this is the end of the first subproblem, this is the end of the second subproblem, this is the end of the third subproblem. Maybe you didn't save those before, but now you just save them along the way, and then you combine them in the end to update your psi with the old psi. So it's also cheap in another way. So if you're doing solving your first subproblem, then you discretize it like this, and then you rewrite it like this. So what you see is the effect of adding the psi is really easy to deal with because it just always groups with the initial condition. So wherever you wrote down the initial condition, you now just add a times tau psi. And so both updating the psi is very straightforward, and dealing with the balancing is very straightforward because you're only changing one term, the initial condition term that you have in your code. And so if you write a code that does the straight operator splitting, then you can easily modify that to include the balanced operator splitting, and we'll do part of that this afternoon. And so that in practice gets you back to the steady state thing. Just wondering what I was going to talk about next year, because I don't remember. Okay, right. So let's sort of recap where we are now with the balanced operator splitting. So we have our equation that we want to solve. We have our delta t time interval and our delta in our h, and we're starting at time tn, having done the previous time interval, and now we're going to take our time step h to tn plus 1 and compute u of tn now with a balanced operator splitting. And in the balanced operator splitting, we break the system into easy to solve subproblems, just like we do for the operator splitting. And then with a balanced one, we just add this one number that we then use to solve in all our subproblems. And then using the balancing guarantees that we now reach the correct steady state. So this is where we are, what we've talked about so far, and then the updating of the psi is very straightforward. All right, so now the next step is to go to the next time step, and what we want to do next is how do we make a dynamic time step so that we're not just using the same h all the time. And that's going to be the next thing that we talk about, and that becomes another aspect of this whole solution scheme, doing dynamic time stepping, where here we're doing small time steps, but here where things aren't changing, where we're near steady state, we're taking large time steps. And so this is exactly why we need the balanced operator splitting, because it's in this part that we want to take as large a time step as possible, and we don't want to worry about that we get to the wrong answer. Because if we didn't, we could wind up here, and then everything else is bad from there. And then we do one delta t, we change what the ion channels are doing, and then do another delta t and go on for the entire simulation, for the rest of the simulation. All right, so dynamic time stepping. This is one of the most important, it's actually probably the most important thing for doing long simulations. Because if you're just doing tiny time steps of, I think they were in that one method section, I think they were talking about like 0.1 microseconds, and you want to do 10 seconds, that is a lot of tiny time steps, and you're wasting a lot of time right here because everything's flat. So there's no point in doing those kinds of tiny time steps. But dynamic time stepping is a whole different animal, and we need to be careful in how we do it. So basically what I just said was h defines how fast the simulation is. If you take a large h, you go very fast, but making a large h can also cause large errors. And that is, of course, what we want to avoid. And making small changes, making small h's when things change is what we want, and then we want to increase the step size when things are not changing rapidly. So it's always a balance of speed versus accuracy. That might be the summation of what simulation is speed versus accuracy. How useful you can be is the speed, and how much accuracy do you have to give up in order to do that. All right, so what's the gist of time stepping? We want to pick a new h. And how do we do that? So what we need is an estimate of the local error. So after I've solved this, the equations over my time interval h, I need an estimate of how good that answer that I got is. Because if that answer is good, and if it's really good, then I probably picked too small an h to begin with, and I could use a larger h for the next thing. But if it's really bad, then if it's bad enough, I actually probably need to do that interval over again. The h that I had picked was too large. And so we need a way to estimate that local error. And so the question is, how accurate is my solution at the end of this h? And then I need a way to turn that error into a time step. And if I have a large error, I want a small time step. And if I have a small error, I want a big time step. So how do we get at this local error estimate? And the way that I chose to do it is a very simple and straightforward way. So we have our basic equation here for our time over our time interval, and what I can do is I can integrate this exactly. So basically by just writing an integral over this. And this isn't particularly helpful, but I can approximate this integral by using the trapezoidal rule. So that I'm using the value at this plus the value of this divided by 2 times the time interval length. So then I have some sort of estimate as to what this should be given these two things. So I have these u's at t n plus 1 on both sides. But, so the key point to this is that these come from the operator splitting. So that's the answer that we got from the operator splitting. This is an exact description of the whole equation that we want to solve, not the bits and pieces the way we chopped it up with the operator splitting. So this is an independent version of this. So if I did a good job, then these two things are equal. If I did a bad job in the operator splitting, then what I get at the end of this calculation will not be the same. Does that make sense? So this is essentially the true solution, and this is the approximate solution that we got. And if I did a good job, then they are the same. But if I didn't do a good job with the operator splitting, if I picked a very large time step, for instance, and didn't solve it very well, then these two sides will not balance, because I solved this in a completely independent way from the way that I solved this. And that's the key to getting an understanding as to how well we're supposed to be doing. This is supposed to be equal to this, but if it isn't, then I didn't do a good job, and that's my error. So the error formula that we're going to use is a relative L infinity norm. So what does that mean? So I'm just taking this equation and moving this stuff over to the other side. So then I'm just subtracting off this side. And then I'm doing a relative error so that I'm normalizing it by something, and I'm normalizing it by the u that I got. So that it's always a fraction of what is wrong, not the actual number. So if I did an absolute error without dividing by this, then if I have like nano or micromolar concentrations at one point, but high concentrations at another point, then I get wildly different sizes in each of these terms from different components, from different boxes. And so by dividing by some sort of normalizing concentration that is representative of the size of the concentrations in that box, I always get something that's between 0 and 1, hopefully, or that it's order 1. And the L infinity norm just means I'm picking the largest absolute value. So for this, that means, because we're using this vector notation, I'm looking at this error in every box for calcium, and I'm looking at it for all the buffers, and then in every box I'm taking the maximum error of this from both the calcium and the buffer, and then I'm taking the maximum over all the boxes. And that's what this means. Is that clear? Yes?
Does this error actually so depends on the value of the age? If you have higher age, then the error will be smaller.
It does depend on the value of h, but it's this h so h comes in two different ways. One, it comes in here, but it also comes into the solution of this. So this one here is just being used to evaluate the integral. So if I have something that is essentially constant, like I'm at steady state, then this integral, I can take h to be very large and still get a good answer because I essentially have a rectangle. So what I'm So if I have something like this, and so if I'm looking here at this, and I'm doing this integral, then it's then doing a trapezoidal rule will give me a very good answer. And so it is this value averaged with this value times h. If I take an even larger interval and do it over this, then I still get a very good answer because it's flat. So in that sense, the h doesn't comes in only when things are changing a lot and you need your trapezoidal rule to give you a good estimate of the area under here for that for this actual integral. Then the h comes into this part, into that part by the operator splitting, and that is then the h comes into play into how wrong your operator splitting answer is. Does that answer your question? Okay. Okay, so then there's always the question of like, what is a large error, what's a small error? And that almost always comes down to a user-defined threshold. So large error, small error. So if my epsilon is bigger than my threshold, then that's a big error. If it's smaller than my threshold, then it's a perfectly fine error. That's almost always the way that it's done. There's some sort of user input as to what is acceptable and what is not acceptable. And then as you decrease the threshold, your error will, overall error should decrease. So now what happens if I have a large error? Then what I'm going to do is, so what happened was that at the end of my calculation, my answer was unacceptably far away from the true answer. So that's unacceptable. So I'm going to do that interval over again. But I'm going to do it with a smaller, with a half a time step. So, so what we just calculated, we're going to do it again, start again here, and do take h half as long. And because operator splitting is consistent, meaning that if I take a smaller h, I will get a smaller error, that should then eventually get us to a place where the error that I'm making is small enough to be acceptable. For a small error, then what I want to do is I want to grow the h. I want to make it larger, and both having the h and using this formula for growing the h, that's on you. There's many different ways to do this. This is the way that I've chosen to do this because for this particular problem it worked well. So what does this mean? So if I pick 1 for h, then 1 to anything is 1, so I will always have the same 1. So if I pick a number that's larger than h, then my h will start to grow, and this is the log 10, is a base 10 log. So if my error is 10 times smaller than my threshold, then this log is 1. And so if I pick h to be like 2, then my h will double in the next time interval. If my error is exactly at theta, then that's log of 1, that's 0, and my h doesn't grow. So my time interval grows depending on how well we did the last time. And this is just one way of doing it. I basically picked a way, found that it worked, and went with it, because you don't want to optimize things, over-optimize. You actually want to do things at the end of the day at some point. But there's many different ways to do this. You don't have to cut the interval in half, but it's dividing by 2 is a good even number. So with this new larger h, we then go on to do the new interval and go on to this interval. So here we went back and recalculated, and then hopefully at the end of that we got a small enough one so that we could then go on to do the next time interval. And so the growth in H is a balance. So you want to maximize H for fast computation. So the larger your H, the more time you can do, the faster you can do it. But large H can also mean large error, which means you might have to recalculate a bunch of intervals. And so you also want to minimize the number of recalculations that you do, because all those, we're using consistency to do this. But anytime you recalculate an interval, that's just wasted time. You learn nothing in that time, and it's just wasted computation time. So you want to both maximize the H, but at the same time not do too much extra work. And that is where this cap H comes in. So I tried different values of cap H for like the growth rate, and then basically just looking at how many calculations did I do and how long did it take, and found that picking H to be 1.5 minimized the amount of time that I took. So here, for a small h, we're not growing fast enough. Our h could have been larger, and we're not doing very many recalculations here, but our h was also not large enough, or as large as it could have been. Here, on the other hand, now h can become very large, but that means I'm making errors in my operator splitting answer, and so I have to recalculate things again. And so over here, we're spending time recalculating intervals, and sort of here in the middle, you're getting that balance as best as possible between large h and not recalculating intervals, which are wasted time. All right, so now we're ready to simulate. We've done all of these things, and we have our new dynamic time step, and we can, we're basically ready to go. So what could possibly go wrong? So we've talked about some of the things that could possibly go wrong. Local error, global error, and stability, and we're probably pretty cool on consistency. And we've also tried to take care of the local error. So the whole point of calculating that epsilon, that error function, was to control the local error. But the question still is that still good enough to not accumulate errors over many h intervals in and a delta t time step? And is controlling that local error good enough to get a correct answer at the end of tens of seconds of simulated time? And that's where checking comes in. Check, 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 do some more checking. And I really can't emphasize this enough. Try to break your simulator. Even if you're working with a package, if you're doing MD or whatever, take a system and really apply extreme conditions and understand what is the kind of answer that you get in that kind of extreme condition, so that you can recognize when accidentally you've done things incorrectly. And this is these two things, and knowing the performance envelope, knowing when your simulator fails, when it gives you a bad answer, are probably the biggest take-homes from this. Because I don't think people do enough checking, and they really hardly ever try to break their simulator. And that's actually when you find really interesting things, but also at the end of the day, if you're a scientist, all you have is your reputation. And checking to make sure that you didn't make a typo or have some other fundamental flaw, and trying to and actually breaking your simulator is what that makes you careful, and that gives you a good reputation. I've asked, you know, in my time about like, you know, can I trust the data coming out of this lab? I've gotten 2 answers. Oh yeah, you can trust anything that comes out of that lab. They're really careful. And the other one was like, you know, they're a little fast and loose. So, you know, you can guess which reputation you probably want. And as a simulator, this is how you do it, by playing with your system. So what do I mean by performance envelope? So this is a great picture for the performance envelope of an airplane. So here's the airspeed, so how fast you're going, and here's how much lift you have. So here you're going up, and here you're diving down. And this is where you want to be. Everything is fine. If you go too slowly, either if you're going up very fast but too slowly, or if you're going down but too slowly, you stall and you might fall out of the sky. The other problem is this part, and I love this picture because it was like structural failure likely, you know, and that is a completely different kind of problem, and that's why doing a lot of checking works, because you have many different kinds of problems that you probably won't even think of that you might generate, but it is actually possible not just to stall, which is something that you know, maybe a lot of people know about, but if you do it wrong, you can actually damage your airplane. And this is I think a really nice sort of way of illustrating that you are also in your simulations looking to figure out what is the green area for your particular simulator, because every single simulator, algorithm, whatnot, has its limits of what it can do. Not everything can do everything. That's pretty self-explanatory. Okay, so in the kind of system that I'm interested in, I did a bunch of checks, and in the workshop later, we're gonna look at some of the kinds of checks that I did. But so I want to go through some of this. So part of the downside of checking is that you need to know what the exact answer is, and you're probably working on something that doesn't have an answer. You don't know what the answer is, otherwise you wouldn't be looking at it, or you wouldn't be computing it. But that actually should not stop you. So first, you can learn a lot by doing simple problems. Not everything, your checking probably should not be on the actual system that you're interested in. It should be on a much smaller scale, and it should involve parameters both that are like what you're doing, but also ones that are wildly different than what you're doing. And by doing a simple problem, you can probably find another method to do it. So, for example, using a tractor, using a Runge-Kutta, a black box out of Mathematica or MATLAB that doesn't take too long to do, even though it's going to be slower than or better be slower than what you programmed, but you can get an answer in a reasonable amount of time. So, for example, so this is the simulation box that I would like to do. Actually, I would like to actually do a bigger one than this, but that's not feasible. And so I do two kinds of I did two kinds of checks. So first was with a really simple system, a 2-compartment system. So here we have 5,000 boxes. What if we just have 2 boxes? And we're going to make it even simpler. So one box has a source and it diffuses into this box, But this box is a bath, meaning that its concentrations never change. So you just have stuff coming in here and diffusing out over here. And that's it. It makes really simple equations. And this is actually the system that we're going to be playing with in the afternoon. And so you might think you can't learn a lot from this kind of system, but that's actually not true. And that's one of the points I would like to make, because that you can use a really simple system to try to understand what can work and what might break. And the philosophy I've always tried to go with is, if you find if it breaks in the simple system, or if it's a problem in the simple system, it's going to be a problem for the more complex system. So you may not find everything, but what you do find will be an issue for you later. The other system that I did is what I call the mini physiological system. So I'm interested in physiology. I'm interested in understanding how this works. I have a particular set of constraints that I have. I have a given set of calcium buffers. There's the fluo-4N, there is the ATP, there's the calmodulin, there's all these biological and experimental ones. Those are set. So now I want to use those, but I want to do it in a system that is not as gargantuan as this one. So these boxes are the boxes that I simulated. There's no others. So again, it doesn't have to be huge, but here we're doing just like 2D diffusion because these are just the boxes. So we have some small boxes, we have some ranitidine receptor-like things here in the blue, and then in the red is where I'm measuring the calcium for my comparison to the Runge-Kutta output, and then these light blue ones are just illustrating the neighbors into which this box diffuses. So it has the major elements of the actual physiological problem that I'm interested in, and this is the other kind of important check. You do something that is similar but smaller than what you're actually interested in. All right, so let's take a look at what I did with the two-compartment model, and this is basically what we're going to be doing in the afternoon. So what we have here is our two equations, and in red I've highlighted all the things that are not the unknowns. So these are generally the inputs to your model. You know what your buffer is, you know what the on rate is, off rate is, you know what the on rate is, you know what your bath concentrations are, you know what your diffusion coefficient is. But from what we're doing right now, from a math and a computational point of view, these are just numbers. I can put in whatever I want. I can put in ones for all of these. I could put in any sort of thing, and I love those kinds of systems because that means I can check a whole bunch of different things. And so we're going to throw out the physical meaning and just use it to test the algorithms. And one of the things I'm particularly interested in is this question of DRD versus RDR. What order do you do your subproblems in? What does it actually matter? And the other question I'm interested in, are there sort of pathological cases that we can see where this algorithm fails? And then do those occur in physiological problems? And we need to be aware of those sorts of cases. So one of the best piece of advice I ever got is, when in doubt, randomize. So I just pick all these numbers randomly and then solve it to steady state with both the operator splitting, the dynamic time stepping operator splitting, and the Runge-Kutta. And then track the error versus the Runge-Kutta, and I did 50,000 different parameter sets. So all of these numbers put together are a parameter set. You do 50,000 of those. And the goal is to basically pick random points in your performance envelope, in your performance graph, and see where the envelope is. So here's a histogram of, so we're looking at the calcium error. So the error in just the calcium versus the Runge-Kutta. And then we did made a histogram of it. So this is the 50,000 different examples with all sorts of random numbers here. And then look at the histogram. So in this slice we had so many different errors, different numbers of times that the error came in here, and then in this slice here, and so on, just making a histogram. So we already see a bunch of interesting stuff in here. So even for this really simple case, order matters. So you get very different histograms for whether you do diffusion, reaction, or whether you do reaction first, then diffusion, and then the reaction. And so you already see really important things coming out of this. And so for example, so here the black being higher than this means that there's a lot higher fraction of these parameter sets had error in this range than they do for the red curve. So that means in general the DRD seems to be more accurate for these kinds of cases. We don't know if that's always true, but in general that seems to be one takeaway. But we also see that in both cases you can get big errors. So here again we're looking at like a fractional error. So this would be like 10% error compared to the true answer. So that's quite a bit. And here we're looking at a few percent error, which is acceptable but not, or maybe acceptable but not great. And so we're already seeing some really interesting stuff, and then the question sort of is, why does that happen? And this is starting to fill out your performance envelope. If we can identify when this happens, we can see what we need to avoid, and we also see that the performance envelope of the order of the two orders are different, and that is a really important just general observation. We're going to explore all this much more in the afternoon workshop, because this is the problem that we're going to be working on and doing exactly this kind of analysis. All right, so we're actually getting towards the end here, so I'm actually going to get to go to lunch pretty early. So for the mini physiological case, I'm interested in just seeing how the system that I'm actually interested in behaves with the dynamic time step operator splitting. So we're simplifying things. It's just 2D diffusion. The boxes don't really care how they're connected, like if it's up or sideways connection. So 2D is fine. And just having a small number of things like this means that the Runge-Kutta can do it efficiently enough. We're using all the physiological and experimental buffers like ATP and calmodulin. And the calcium concentrations are physiological, and we have ranitidine receptors that open randomly, and they make a physiological level of current. I did try to make it a little bit extra difficult. So the dyadic cleft is like 12 nanometers tall, and so in this case I made all these boxes just 1 nanometer tall. So if I have flux going into a box, it's a much smaller box, so it makes a very high concentration very quickly than it does in a bigger box. It makes it more numerically challenging. So you have to have like smaller time steps to get sort of relatively small changes in the box concentration. So, but that's what we worked on, the system that we worked on, and we found some interesting things. So here I have basically the curves that I've shown you a couple of times. So here we have our calcium concentration at that red spot that I showed you on the thing previously, and just looking at it going forward in time, and actually running like actual timescales that we're interested in. And so we see a couple of really interesting things. So the first thing that we see is the thing that should jump out at you is this red line here. It is not the right answer. So what is that? That is the RDR. So again, order matters. But what is interesting here is after all the analysis that I did, I figured out that the culprit was the ATP. If I didn't have the ATP, the RDR still isn't perfect, but it's a lot better. And in both cases, the DRD works really well, but the RDR, especially with ATP, is highly problematic. So it's not something that we want to use. I don't know if I want to go to lunch anymore. But so what the heck is going on? So then we also looked at stability. So here we have 10 of these delta T intervals. And here we're trying to see, is the algorithm stable? And so here we did 2,000 delta T intervals. And we're looking at the error compared to the RK, and that is not drifting off. So it's very stable. So we do see some things here which would look like they're problematic. So you have to investigate those too. Why are those error spikes so large? What's going on there? So we audited all of them. We looked at all of those, and what it turns out what happened here was that you have a part of the system or a time in the system when the buffer, there's so much calcium that the buffer concentration is really low. It's almost zero. And so when you're doing a relative error, if you have two small numbers and you're off by a little bit and you're dividing by a small number, suddenly you can get a large looking relative error. Does that make sense? And so what happened there is that the buffer concentrations got so low that compared to the RK solution, if it was 10 nanomolar and the RK said it was 1 nanomolar, well then you're a factor 10 off. But you're a factor 10 off times 0, and so it's not biologically relevant that the concentration is 10 nanomolar or 1 nanomolar. And it's also not even clear that the RK is the true solution then, because then it might be running into problems as well, trying to make that kind of small solution. And if you look at the RK solution for some of the small parameters, it wiggles around when the concentration is like nanomolar. So it's not actually probably the correct answer. So, but it is important to go in and look at these cases and convince yourself, well, actually that's a bad way to put it, and prove to yourself that it is a case that is not relevant. If it is a relevant case, then you have to address it. Then it's probably your algorithm. And in this case, it's not relevant because the concentrations of buffer being that low doesn't matter for the physiological case. And so the one other important reason that I would like to point out to check is that if you didn't know at the beginning that operator splitting preserves or does not preserve steady state, that it doesn't give you the correct steady state answer, this is where you would find it. Because it was, I came across this fact by accident. It was before I did this, but here I would have really found that something was seriously wrong. And then I would have probably spent a great deal of time figuring out where the bug in my program was. And at some point later, with a lot of work, probably figure out that it was the algorithm itself. And this is the kind of stuff that you need to be careful about, because operator splitting is old. It goes back to the 60s. It's very simple to implement. And it's got the mathematical stamp of approval that it's accurate, it's consistent, but that means you have to have small h. And if you don't have small h, those things can accumulate, and you're going to get problems. So that's the other reason, because there might be something that you have done that is incorrect, or the algorithm is not complete. All right, so what have we learned from this mini physiological case? Again, order matters. And in this case, it matters a great deal, because ATP is problematic, and ATP is ubiquitous in biology. So what is special about ATP? I mean, if you know anything about biology, you know that it's the energy molecule. It's what is broken down to get energy. And so it is there everywhere. It also just happens to bind calcium. And so we have to include it in our thing here. But it's also different than the other proteins in that buffer calcium, in that it is there at really high concentrations. It's like there, like 5 millimolar. So the calcium concentration is usually say 0.1 to 100 micromolar. So this one is present at 5,000 micromolar. So there's a huge amount of it. It doesn't buffer calcium very well. But because there's so much of it and there's so little calcium, it has a noticeable effect. Like I showed you those two curves, they were different with or without ATP. So while one molecule by itself doesn't do much, because it's actually sort of a bad calcium binder, there's so much of it that it has an impact. And so that means that basically we have to use DRD and not touch this. But without doing all this checking, without asking the question, does order matter, we could have run into some serious problems. And we'll talk a little bit in the second session as to why ATP is problematic, or why it falls into a general class for which the dynamic time stepping operator splitting doesn't seem to work well with at least RDR and sometimes DRD. All right, so it's going to finish off before we go to lunch with the slide I started on. And this is starting with know the biology. Like now, what we just talked about with the ATP is knowing the biology. What is different about ATP sort of physically that might be the cause of the problem? And also just knowing what is acceptable or what is normal. And we saw that how to tailor the specifics of our system of equations allowed us to do a more efficient solution of each of the subproblems, especially the reaction subproblem, which is where the difficult part lies, which is where the nonlinearity lies. And you can probably make a Ferrari to do this, but I don't think you really need one. Our Porsche seems to have done pretty well. And the reason that I say do better than a Honda, as a professional simulator, you should be able to make a Honda with very little effort. And so try harder. And then check, check, check. Even on simple problems, you can get a lot of information as to what might be wrong, what might go wrong, and then know the performance envelope of your simulator. Because the one thought I want to leave you with is I can virtually guarantee you that if you don't know the performance envelope of your simulator, one day you will accidentally wind up in one of these red spots and not know that you have a bad answer. And that is what will be problematic for you personally, because that damages your reputation.